Learning outcomes. Dear students, after studying this module, you shall be able to know about principle and basics of electrophoresis and buffer systems, learn about the gel electrophoresis, its role in resolving nucleic acids and proteins, its modifications and staining procedures. Evaluate the analytical difference between resolution of nucleic acids and proteins. Now students, let us start with the introduction of this module. Electrophoresis is an electrokinetic phenomenon which was first observed in 1807 by Rius of Moscow State University, who noticed that clay particles dispersed in water migrate upon application of a constant electric field. Electrophoresis, also called as cataphoresis, is caused by the presence of a charged interface between the surface of a particle and the surrounding fluid. Alternatively, the term electrophoresis can also be defined as the motion of particles dispersed in fluid under the influence of a spatially uniform electric field. Electrophoresis is one of the major technologies currently available for molecular separation in laboratory. Apart from many different physical arrangements for the apparatus and regardless of the medium through which molecules are allowed to migrate, all electrophoretic separations depend upon the charge distribution of the molecules being separated. Because of its ease, inexpensiveness and high resolving power, it has become quintessential in molecular biology laboratories. Electrophoresis can be performed in one plane, that is one-dimensional separation, or two phases, that is two-dimensional separation. One-dimensional electrophoresis is more commonly used for separation of proteins and nucleic acids such as DNA and RNA. Two-dimensional electrophoresis involves separation of proteins using two physicochemical properties of proteins. It is extremely accurate in resolving all of the proteins present within a given sample and has very high resolution, which is still unchallenged after almost 40 years of its discovery. The support medium for electrophoresis can be performed into a gel within a tube, or it can be casted as a flat sheet into layered structures. Sheets provide a larger surface area for resolving a number of samples alongside and making their simultaneous comparison with one another. In addition, layered gels are used in two-dimensional separations, while tube gels are used for easy one-dimensional separation. Next, we will be studying about gel electrophoresis. Gel electrophoresis refers to using a gel as the sieving medium during electrophoresis, which retards the migration of charged molecules. The main function of the gels is to subdue the thermal convection resulted by the electric field. It is commonly used for separation of biological macromolecules such as deoxyribonucleic acid or DNA, ribonucleic acid or RNA, and proteins as well as nanoparticles. Gel electrophoresis is usually performed for its downstream analytical applications such as visualization of nucleic acids, RNA and DNA, and in particular amplified DNA following PCR, RFLP, cloning, DNA sequencing, or southern blotting and northern blotting for further characterization. Agarose based gels are commonly used for most of these electrophoretic applications. Next is agarose gel. Agar is a natural polymer which is derived from seaweeds, Gelidium and Gracilaria species. The purified form of agar is known as agarose, which is extensively used in electrophoretic separations of nucleic acids. Agarose is a biopolymer, a linear polysaccharide which can be melted to cast gels. Agarose upon melting in aqueous phases becomes gelatinous and is used because of its sieving effect in electrophoresis. Agarose gels are suitable for resolving extremely high molecular weight nucleic acids above 200 kilodaltons. In addition to using pure agarose gels, acrylamide concentration of 0.5% can be used along with agarose and is particularly useful for the separation of large sequences of DNA or other nucleic acids with molecular weights of up to 3500 kilodalton can be separated. Agarose acrylamide gels are extensively used for the determination of gene maps. However, if acrylamide is used alone, its concentration needs to be reduced to a level where handling gels becomes impossible as it would not solidify. The next type is acrylamide gels. In case of proteins, the pore size of agarose gel does not offer desired level of resolution and hence polymeric acrylamide gels are used. Anionic detergent SDS, which is also known as sodium dodecyl sulfate, is commonly used with proteins, imparting proteins with negative charge when attached to the SDS anions. This technique was discovered by Lamelli and is collectively called SDS PAGE, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis, and is used to compute the molecular weight of proteins. Polyacrylamide gels are formed from the polymerization of two compounds, that is acrylamide and N,N-methylene bisacrylamide. For the gels, bis in acrylamide acts as a cross-linking agent. The polymerization is initiated by the addition of ammonium persulfate, that is APS, along with N,N,N,N tetramethylene diamine, that is TEMED, that act as a catalyst. These gels are hydrophilic in nature, neutral, and are formed of long chains of hydrocarbons cross-linked by methylene groups and having a 3D network. Here in this reaction, we have shown the structure of acrylamide monomers and the polymerization process. In this diagram, the acrylamide monomers cross-link to form branched or cross-linked polymers. Separation of molecules is based on the pore size of the polyacrylamide gel, and two factors determine the pore size. The first factor is the total amount of acrylamide present, that is %T, and the second one is amount of cross-linker, that is %C. There is a decrease in the size of pores with the increase in amount of total acrylamide present. The size of the pores will increase with increase or decrease in %C, and cross-linking results in the smallest size of pores with 5% C. Also, these gels are denoted by % solutions and will have two necessary parameters. The total acrylamide is given as % of weight by volume of the acrylamide plus the bisacrylamide. Thus, a 10% T would indicate that there is a total of 10 grams of acrylamide and bisacrylamide per 100 ml of gel. A gel designated as 10% T, 2.6% C would have a total of 10% weight by volume acrylamide in addition with bisacrylamide, and the bisacrylamide would be 2.6% of the total. Higher the gel concentration, the smaller the pore size of the gel and the better resolution it provides in the separation of smaller molecules. The % of gel to be used depends on the molecular weight of the protein to be separated. Next, we will be studying about the cationic versus the anionic systems. The proteins are separated based on their charges, which may be either negative or positive depending upon the pH of the buffer. Under normal operation, a gel column is partitioned into three sections known as stacking gel, resolving gel and the sample gel. the sample gel may be eliminated and the sample is introduced via a dense non-convective medium such as glycerol or sucrose. Electric current is passed through the gel with the help of electrodes, that is anode and cathode, attached at both the ends of the terminal. If the arrangement of electrodes is in such a way that upper bath is cathode, which is negatively charged, while the lower bath is anode, which is positively charged, and anions are allowed to flow towards the anode, then this type is called as anionic system. When the flow is in reverse direction, that is cations flowing towards cathode, which is the negatively charged electrode, is called a cationic system. Next, we will be studying about tube and slab gel systems. Gel electrophoresis is based on two approaches. One, column electrophoresis, which uses tubular gels casted in glass tubes as shown in this figure, while the other uses slab gel as we have shown in this figure and uses flat gels formed between two glass plates. Tube gels are advantageous as the movement of molecules through the gels is less prone to lateral movement and thus offers slightly improved resolution of the proteins. These gels are commonly used for isoelectric focusing, or better known as IEF, of proteins, which is often used as the preliminary step for the two-dimensional electrophoresis. The polymerizing material used for isoelectric focusing can be either acrylamide at low concentration or agarose depending upon the downstream application. Here we have again shown the tube gel system. Gel is casted within the capillary tubes seen in the system and is used for electrophoresis. This figure shows the slab gel system, a vertical slab gel electrophoresis unit that uses acrylamide gels for resolving proteins in the sample. Multi-wells allow simultaneous analysis of a number of samples. Also, the main advantage of the gel slab is that it can be used for running multiple samples in the same slab. Slab gels are casted with multiple well setups such as the samples run in parallel lanes, size and number of which can be varied within a limit. Since the samples run in the same medium, it minimizes variation due to minor changes in the gel structure and buffer concentration. This property of slab gels makes this a standard technique of choice for any blot analysis, nucleic acid analysis as well as autoradiography. The theory and operation of slab gel electrophoresis is identical to the tube gel electrophoresis, but have gradually replaced the tube gels because of multi-sample analysis they offer. Both slab gels as well as tube gels can be easily casted in-house from monomeric materials. Next, we will be studying about continuous and discontinuous gel systems. With the introduction of gel electrophoresis, the use of gels was limited to separation of components using a single gel with uniform pH throughout. Using this system, molecules were separated on the basis of their mobility through gel matrix. Over the time, this system has been replaced with discontinuous multiple gel systems where a sticking gel augments a separating or a resolving gel. These gels have different concentrations of the same support media and pH. The pH of stacking gel is 1 to 2 units lower in comparison to resolving gel. This difference in concentration and pH determines how the molecules separate when they enter the separating gel. Stacking gel is cast over the resolving gel. The volume of the sample that is to be applied on this gel, its height is kept lower than the height of the gel slab, that is based on the technique of isotachophoresis. In this figure, we have shown the mechanism of isotachophoresis and resolution using discontinuous gel. The chloride ions and glycinate from the resolving buffer tris-glycine vary in their migration rates, and leading ion is usually the chloride ion, while glycinate is the trailing ion. Chloride ion enter from stacking to the resolving gel first and rapidly moves down the gel, followed by the proteins and glycinate ions. The glycinate ions overtake the proteins and give rise to uniform linear voltage gradient, within which the proteins sort according to their molecular weight. It means the sorting is done according to their molecular weight. The proteins in the sample gel will concentrate into a small zone in the stacking gel before entering the separating gel. The concentrated bands of proteins migrate through the separating or the resolving gel in form of narrow concentrated bands that are then separated from each other on the basis of molecular weight on the separating gel. The separation of the protein bands is boosted as they enter the separating gel and the ions pass in pairs through the gel column. Each ion in the pair has the same charge polarity as the protein, usually negative, but differ in charge magnitude. One ion will have a much greater charge magnitude than the proteins, while the other has a lesser charge magnitude than the proteins. This ion will move faster through the gel, that is, the one that will have the greater charge and is known as the leading ion, while the trailing ion is that ion which has the lesser amount of charge. When an anionic system is employed, the chloride ions and glycinate ions are derived from the reservoir buffer of tris-glycine. Generally, chloride acts as the leading ion and the trailing ion is the glycinate ion. The chloride ions are the first one to enter the separating gel, after which comes the proteins and finally the glycinate ions. Proteins are surpassed by the glycinate ions and thus help in establishing a uniform linear voltage gradient in the gel. The proteins then sort themselves within this gradient according to their molecular weights. Next, we will be studying about resolving buffers. Choice of buffer system for an electrophoresis experiment depends upon number of factors such as type of gel, molecules to be resolved and the characteristics of molecules to be resolved. Most commonly resolved biomolecules are nucleic acids and proteins, which are resolved on acrylamide gels and agarose gels. Here, first one we will be studying about acrylamide gels for resolving proteins. The sharpness of the bands in the gel can be augmented by employing a discontinuous buffer system for the protein separation. A gradient of ion is created during the process of electrophoresis in the discontinuous buffer system in the stacking gel, which has low acrylamide concentration at an early stage of electrophoresis. This results in stacking of proteins into a single sharp band. The proteins are separated depending on their molecular weight and by the sieving property of the gel that exclude the ion gradient, and also the negative ions present in the buffer system outrun the SDS covered protein stack. As a common practice, tris-glycine or lamellae buffering system is used with reduced protein samples on discontinuous gels that stack at a pH of 6.8 and resolve at a pH of approximately 8.3 to 9. The values of pH mentioned helps in the formation of disulfide bonds among cysteine residues, particularly when they have high concentration, as the pKa of cysteine ranges from 8 to 9 and the reducing agent present in the loading buffer does not co-migrate with the protein. Inclusion of reducing agent, example sodium bisulfide, that move into the gel ahead of the proteins is done to maintain a reducing environment further aids in resolution. With further development in buffer technology, new and more efficient buffer systems have been explored and established, such as Tricene Tris, MOPS and more. These further enhance the resolution of lowering down the limit of stacking using the same gels, which in turn enhance the resolution of lower molecular weight proteins and peptides without using more concentrated acrylamide gels. Next is agarose gels for resolving nucleic acids. TAE, that is tris acetate EDTA buffer, is a buffer solution containing a mixture of tris base, acetic acid and EDTA, and is used to resolve the nucleic acids on agarose gels. It is made up of tris acetate buffer, usually at a pH of 8, and EDTA, which removes the divalent cations. Another commonly used buffer system is TBE, which is made from tris base, boric acid and EDTA. TAE has a lower buffer capacity than TBE and can easily become exhausted, but linear double-stranded DNA runs faster in TAE only. Here in this figure we have shown the agarose gel electrophoresis. PCR products are resolved on 1% agarose gel and visualized by ethidium bromide staining. First lane is loaded with the standard molecular weight marker. TAE buffer is commonly prepared as a concentrated 50x stock solution for laboratory use that can be prepared by dissolving 242 grams of tris base in water, then by adding 57.1 ml of glacial acetic acid and further 100 ml of 500 millimolars of EDTA of around pH 8 solution, and then bringing the final volume up to 1 liter. This stock solution is then diluted in the ratio of 50:1 with water, so to make 1x working solution that contains 40 millimoles of tris, 20 millimoles of acetic acid and 1 millimole of EDTA. TAE buffer is used as both a running buffer and is used in casting of agarose gel. Compared with TBE buffer, TAE buffer offers advantages in subsequent enzymatic applications for the DNA sample, which are inhibited by presence of boric acid in TBA buffer. Next is visualization and storage of gels. After the completion of electrophoresis, the resolved molecules in the gels can be visualized by staining. Ethidium bromide is an intercalating agent that is used to visualize DNA or RNA. On the other hand, proteins can be visualized by staining with Comasi brilliant blue dye or with silver stain for high sensitivity. Other methods may also be used to visualize the separation of the mixture's components on the gel. If the analyte molecules fluoresce under ultraviolet light, a photograph can be taken of the gel under ultraviolet lighting conditions, often using a gel documentation system. If the molecules to be separated contain radioactivity added for visibility, an autoradiogram can be recorded of the gel. Multiple samples loaded on the same gel will resolve into parallel lanes running adjacently. Depending on the number of different molecules, each lane shows separation of the components from the original mixture as one or more distinct bands, that is one band per component. Incomplete separation of the component can lead to overlapping bands or to indistinguishable smears representing multiple unresolved components. As a common standard practice, the analytes are resolved on the gel along with standard molecular weight markers that are commercially available. If such a marker was run on one lane in the gel parallel to the unknown samples, the bands observed can be compared to those of the unknown in order to determine their size. bands in different lanes that end up at the same distance from top contain molecules that passed through the gel with the same speed, usually means that they are approximately the same size. The distance a band travels is approximately inversely proportional to the logarithm of the molecular weight of the molecule. Now, dear students, let us summarize this chapter. Electrophoresis is an electrokinetic phenomenon which is used as one of the major technologies currently available for molecular separation of charged biomolecules in laboratory. Most commonly used form of electrophoresis is gel electrophoresis, where a gel based matrix such as agarose or acrylamide is used for separation of molecules. Gels are used as a sieving medium during electrophoresis that retards the migration of charged molecules. For resolving nucleic acids such as DNA and RNA, agarose gels are commonly used. Agarose is a biopolymer which is derived from seaweed species, example Gelidium. Acrylamide gels are polymerized by cross-linking at a desired percentage of cross-linker and are used for the resolving proteins. In addition to choice of sieving gels, buffers and other factors also influence the migration and resolution of biomolecules by electrophoresis. Commonly used buffers for resolving nucleic acids are TAE and TBE, whereas the separating proteins, a SDS page is used, that is a discontinuous gel system is preferred with tris-glycine or tris-trycine buffers along with SDS. Resolved biomolecules can be visualized by staining them with a suitable compound. Ethidium bromide is commonly used to visualize DNA and RNA. Kumasi brilliant blue or silver staining is also used to visualize the proteins.